Okay, let's get going on chapter 4 here. So chapter 4 is prokaryotic anatomy, basically. Your book just titles it Bacteria and Archaea, although they don't talk to you very much about archaea. And I like to think of it as just sort of a general walk through all of the features that you can find in bacteria. Some are going to be the same, and you're going to recognize them from eukaryotic cells, and some of them are going to be new and different, and you're just going to be like, oh, I've never heard of this before. Sorry about that. All right, so first off, I think it's really important at this juncture for us to, why that's so big, to be really clear about what we're talking about. We are talking about prokaryotic cell or prokaryotes. And remember, this is a cell type that is really important, and only bacteria and archaea, those are the two domains, so big, large groups of living things that are prokaryotes. You are a eukaryote, and you, animals, plants, fungus, and protists all go in the eukaryotic category. So I would remind, it's probably a good time, and it looks like I'm out of practice here and I'm writing too large, so I'm going to try to write a little bit smaller, because I think it's a good time for us to make a comparison between prokaryotes and eukaryotes, because this is going to continue to haunt you all of your time that you study biology. So prokaryotes, can anybody, either in the chat or if you want to join in, can anybody tell me an example or one characteristic of prokaryotes that's different or similar in eukaryotes? No nucleus, says Bridget. Okay, so I'm guessing that you're going to say that you're saying prokaryotes have no nucleus. And so we very specifically, because now we're in a 200 level class, so we're going to use more sophisticated language than just no nucleus. Okay, we want to say lack membrane bound nucleus. And I would remind you from Bio1 that membrane is a phospholipid bilayer, and that's really important because we're going to see that prokaryotes have what we refer to as a nuclear area, but there's no membrane around it. And eukaryotes, we would say membrane bound nucleus present. Okay, so that's one thing. That's great. Anyone else have anything else that they would care to add to that? Any other characteristics that we know are different in prokaryotes and eukaryotes? All right, then I'll just add to the list. Not only do prokaryotes lack a membrane-bound nucleus, they also lack any other membrane-bound organelles. So be careful here, right? Membrane-bound organelles. So that means they don't have a chloroplast, they don't have mitochondria, they don't have endoplasmic reticulum, but it doesn't mean that they can't make proteins or that they can't turn light into ATP, right? They just don't have that capacity in the form of a membrane-bound organelle. And of course, eukaryotes have membrane-bound organelles. Like the mitochondria and the chloroplast, right? Those are probably the two big ones that everyone thinks of right off the bat. I'm checking to see if anybody else, no one else has volunteered anything. After that, after the presence of a nucleus, a membrane-bound nucleus, and the presence of membrane-bound organelles, there are several other things that differentiate prokaryotes from eukaryotes, particularly that they have a single chromosome. One chromosome. It is constructed of DNA, of course, and DNA, of course, is a double helix, and it is double-stranded, D for double, right? But it is arranged, the bacterial chromosome is arranged in a circle. So you'll see that as the semester goes on, oftentimes I'll draw the bacteria kind of looking like that, and then I'll say, and here's the DNA, and I'll do this. I'll kind of make it like, so it looks like it's double-stranded, and sometimes if I'm feeling particularly artistic, I'll put little lines in there to indicate it's a double helix, right? Eukaryotes, as you may be well aware, they have multiple, meaning more than one, linear chromosomes. Your chromosomes, right? So for example, if you are a human, you would in your cells possess 46 chromosomes totally, right? You have 22 pairs of chromosomes with a pair, and then a pair of, we call them autosomes, with a pair of sex chromosomes making up that last pair. So that's a big difference. Bacteria have much less genetic information than eukaryotes. However, they are capable of doing all the things that are necessary inside a cell to be considered a living thing. Another major difference that people often bring up is the size, right? Prokaryotes tend to be about 10 times smaller than eukaryotes. So I'm going to write that, 10 times smaller than eukaryotes. And we alluded to that a little bit on our last meeting on Monday when we were looking at the sizes of things in a microscope and what we could see with a compound bright field microscope compared to an electron microscope. And if you are talking about specific measurements, we're talking about a prokaryotic cell, an individual prokaryotic cell between 1 micrometer and about 10 micrometers at the largest end, and eukaryotes, as I mentioned, about 10 times larger than that. So 10 micrometers, that's the size of a red blood cell, up to, at their very largest, 100 micrometers. And some of those are single cell protist, or a piece of a fungus that's very large. It's not super common. Now, and probably the last important thing that I would put on this list is that prokaryotes do not reproduce by mitosis and meiosis. Their reproduction is predominantly, we say, asexual, because we are not mixing haploid cells between two organisms. And the process is actually known as binary fission. In eukaryotes, of course, the reproduction can be asexual, so where we make an exact copy of a cell and we have a name for that. We call it mitosis. Those of you that remember from Bio 1, I think that was chapter 12. And we talked about sexual reproduction, right? Particularly concerning haploid sex cells or gametes that are produced by meiosis. I know that's very tiny there at the bottom, but hopefully that's all those things you can remember from Bio 1. So 1, 2, 3, 4, 5. Those are 5 major differences between prokaryotes and eukaryotes. Now, this semester, we are mostly focusing on prokaryotes, which is why all of chapter 4 is going to be about these things, plus any extra unique features that bacteria have. And chapter 5 is going to be all about eukaryotes, okay? The ones that we're going to study in microbiology. We're not going to talk about animals, and we're not going to talk about plants, but we may talk about algae and other protists, and of course fungus in chapter 5. Okay, chapter 5, it should mostly be review for you, especially the beginning part. So all the list of all the organelles and their functions, I'm going to ask you to review that part on your own. And on Monday, I'll just be going through the part of chapter 5 that is probably new or newer for most of you, meaning fungus, protists, fungus, and some of the parasitic worms, which actually are animals. If I remember correctly, I think when I go to a new screen, this one, I lose this one, but I'm not sure. No, maybe I can go back to it. Oh, it's still there. Okay. All right, so now I'm going to go to a new screen, and we're going to start talking about the bacteria anatomy in particular. Okay. So there is a lovely picture in your book. It's on page 83 in the new version of the textbook. It is figure 4.1. So if you're following along at home, or if you're using those old PowerPoints, it's probably close to this. And it's a picture that takes up almost the whole page, and it's a picture of this giant bacteria like this. I'm gonna try to redraw it as best I can. And it has all of these extra layers outside. So we have the cell membrane, we have the cell wall. I don't know if I'm gonna be able to do this. The cell wall red, and then we have, sometimes we also have a S layer or a capsule or a glycocalyx outside of that. And then of course it shows all of the features inside. So it shows this big area of nuclear DNA. And notice it's not a perfect circle as I drew it before, and there'll be ribosomes in here and some other stuff, inclusion bodies, maybe a few inclusion bodies. I'll put a few of those in there. Ribosomes, remember, have 2 parts, the small subunit and the large subunit. They're for protein synthesis. We'll talk more about those in a second. And we're going to go through, and then there are some extensions to this cell. One of them, super important, is a flagella. It's a sometimes. Some cells have them, some don't. And also, I think they show fimbriae coming out of one end of the bacteria. But usually if a bacteria has fimbriae, they're all over the cell. And so what we're going to do in the next couple of panels here, we're going to start on the outside. And I think I'm actually going to start with the flagella, because that's what your book does. And we're just going to march through these from the outside of the cell to the inside of the cell. And we're going to go through things that are different from eukaryotic cells and things that are unique and the parts that I think are important. If you're following along in your book, you'll see that I can't go into as much detail as in your book. The things I highlight are certainly the things that I am going to expect you to remember. Okay, so I'm going to leave this guy here and add another plain board and start on the outside of the cell, which your book talks about first about the bacterial morphology, which is fine. I'm going to do that first because they've moved it. If you're looking at the notes from chapter 4, this morphology and arrangement is actually at the very end of those notes. But in the new version of the book, they've moved it up to the front of the chapter. So I'm going to try to accommodate that and move it up to the top of the chapter. So morphology is a fancy word for shape, right? And arrangement is like it sounds, but we're talking specifically how an individual bacteria, an individual bacterial cell, how it is located relative to its neighbors, neighboring cells. And what's different in some ways about bacteria than in eukaryotes. I know in eukaryotes you think about cells being in tissues, and those would be their neighbors, right? And there are things like if you're taking AMP, right? There's different tissue arrangements. If the cells are in a column, we call them palisade cells, right? In bacteria, though, bacteria tend to be sticky, and usually when they divide, sometimes when they divide, they stay hooked together like this. And as they continue to divide, which remember might be every 20 minutes, they stay in a clump or a cluster. So these arrangements are going to be specifically for bacteria that stay near each other or attached to each other after cell division. Okay, there are 3 and only 3 shapes that you're going to see, and we're going to focus on during the course of the semester. And those shapes are, we call them coccus, which is another word for sphere. But on lab reports, if I ask you to report the shape of the bacteria and it is round or a sphere, I want you to say coccus. I want you to use the scientific terminology, okay? And they show some pictures of cocci, right? That's would be the plural. So we would have, if they're single cells, they look like this. And again, they're similar to what I drew for that first arrangement there. If the coccus are arranged in a clump, right? So coccus is the shape. If they're arranged in a clump like this, or a cluster, we say, then their arrangement, the coccus, the morphology, gets a prefix, and that prefix is Staphylococcus. Okay. They're not just staphylo, that isn't a word by itself, it's just a prefix. Okay, so that's shape 1, coccus. If the shape is round or sphere, but they're arranged in a chain like this, right? If that's the arrangement, then we call that something else. We call that Streptococcus. This is just a description. This is not describing the bacteria. This is not describing the bacteria that gives you a strep throat. This is just describing any coccus bacteria that is arranged in a chain when we looked at it under the microscope. Notice how this is a lowercase s and this is a lowercase s. Our second shape, so I'm going to erase this. Our second shape is bacillus. And bacillus refers to rod-shaped. Okay. And again, the plural would be bacilli, right? And these can be clear rods like this, like is how I usually draw them, like a little capsule, like or a medication that you might take. But sometimes they're very tiny rods like this. We would almost call them a cocobacillus. Right? But if the bacteria is rod-shaped, we refer to it as a bacillus. And that would be the morphology, the shape. The arrangement of bacilli usually occurs as single. That's pretty self-explanatory. Sometimes they're arranged like this in a pair. We would call those diplobacilli. And occasionally they're arranged in a chain. This maybe. Right. And then we would call them, we'd use that same prefix, streptobacilli. Bacillus, if they were talking about an individual cell. Because of their rod shape, they usually don't occur in clumps. So there's no such thing as a staphylobacillus. Okay. And then the third shape is curved, your book is calling it. And that encompasses 3 main subcategories, curved. The first one we refer to as Vibrio, with a small v, because again, I'm not describing the bacteria that gives you cholera. Right? Vibrio in shape is kind of comma shaped. Kind of looks like a little comma. Spirillum, I guess, right? That looks like a spiral. Usually has 1 or more turns to it. These are also usually very thin and faint and wispy. So if you look at them under the microscope, it's almost like looking at spaghetti. They're so small and thin, like angel hair pasta. And the third type of curved are spirochetes. And those are actually complete corkscrews. So if you look at them three-dimensionally, they look sort of like what I just drew there. Or if you look at them under a light microscope, they actually kind of look like this, like a zigzag, because that's the best resolution you can get, right? Lyme's disease, the organism that causes Lyme's disease is a spirochete, for example, right? But its name doesn't have spirochete in it, unlike these guys, where Staphylococcus and Streptococcus. So morphology is just the shape, sphere, rod, sphere, coccus, bacillus are curved, and the arrangement is going to be single, or in the case of coccus, clumps or clusters, which will be Staphylococcus, chains, which would be Streptococcus. The bacilli are usually single, occasionally diplo, and the curved, these are all single in their arrangement. Why is that important? Well, it's important because when you look at them under the microscope, this is what you need to record. And sometimes just the morphology and the arrangement gives you a lot of information about what bacteria it might be. And it helps you determine what step you would do next in the inspection, or what tests you would set up for identification, thinking of those 5 eyes again. All right. Now let's go back and start a new screen, and let's start some of the prokaryotic structures. And the first structure that we're going to talk about, on the farthest most outside of the bacteria possible, would be the flagella. Okay, the structure of flagella is very different in bacteria than it is in eukaryotes. The structure of the flagella, it has 3 major parts. We refer to them as the basal body, the hook, and the filament. The hook is like it sounds, it's the part that's curved, and the filament is the part that sticks out of the organism. The basal body is what attaches the flagella through the cell wall of the organism. And the filament is what actually spins, right? It actually causes the motility. So the function of the flagella, of course, is to move the bacteria. Oh, I didn't spell that right. Motility, there we go. We don't say locomotion. That's a 7th grade high school word for the function, motility. And they'll ask you that sometimes in the virtual lab when you're doing an experiment. They'll say, is this bacteria motile? Which is a version of that word, right? Does it have a flagella or some way to move? Now, these flagella can be arranged just like our cells. They can have different arrangements. They can be arranged singly, which we refer to as monotrichous, or they can be arranged, you can have 2, one on either end or even on the same pole, and we refer to 2, I believe, is amphi. Wait a minute. No, amphi's a clump on one end. Lopho, I think, is 1 on either end. And then there are some that are peritrichous, which means they have flagella all the way around the whole organism. So let's take a quick look here. Mono, Lopho, amphi, and peri. Again, why do we care? Because if we stain with a flagellar stain, or we look at a cell that has flagella, you'll be asked to identify what the arrangement of the flagella is, because again, that's going to give us clues as to what bacteria this is specifically, because there are only a very few that are peritrichous. So LOFO is a clump of flagella, more than one on one end. It's usually on one pole, we'll say, on a pole. Amphi is one on each end, 1 to 2 on each end or pole of the cell. And peri is all around, like this. Proteus is peritrachus. It looks like that, like a little, almost like a little bug. Amphi, about 1, maybe 2 on each end like that. LOFO looks kind of like this. There's a bunch all on one pole, and obviously a single. I don't have to draw that for you. Cool things about flagella: when they actually are engaged and the organism is using their flagella, we refer to it as the motility as a run and tumble. Because if you watch them, and there's probably a video that I can post for this, if you watch them, it looks like it's random movement, that they move in a straight line for a while, and then they kind of flop all over the place, and then they move in another straight line. The straights would be the run, the flopping around would be a tumble. And originally people thought for many, many years that the tumbling was completely random. It's not. It's actually, the bacteria is actually sampling the environment. So the bacteria actually is sampling the environment and moving toward food or concentrations of sugars or proteins and away from concentrations of toxins or antibiotics. So this movement is actually directed. It's actually, believe it or not, even though the bacteria don't have a brain, they can sense concentration gradients and they can move toward positive, like food sources, and away from things that might be detrimental to them. All right, so that's our first attachment. I should mention that the flagella is made out of a protein called flagellin. That's the protein, the main protein. So it doesn't have microtubules in it like a eukaryotic flagella would. All right, second, I think I'm just going to erase this board. Second structure that we need to talk about is a filament. In our bacteria that I drew on the first page, or that you'll find in your book as figure 4.1, I like to call that bacteria superbacteria because it has every single unique structure. Of course, there aren't really any bacteria like that. All right, so the next one is a fimbriae. So if we go back to this picture that I had weakly drawn on the whiteboard here, this is a flagella, which we've discussed. These are the fimbriae that are sticking out. They can also be arranged on one end, but more commonly they're smaller than flagella and more numerous and considerably shorter, and they're made out of a different protein, which is probably why they get their own category. They're made from a protein we call pillin. And if there are 1 or 2 of them, that's what we actually call them, pilli or a pillus. So if you remember from Bio1 from conjugation, that's what that's for. It's for exchanging DNA. Many, we refer to them as fimbriae, and they have 2 functions. One is attachment. We call that adherence because they're sticky. Proteins are sticky. It helps bacteria stick to places that are inhospitable to them. And the other function, they can actually also be used for motility. And that's a recently discovered function, that fimbriae are actually used for motility. We didn't think that for a long time. So I don't think if I want to add anything else about fimbriae. I don't think so. Fimbriae and flagella both are a sometimes, meaning some bacteria have them. Most bacteria do not. Most of the bacteria that possess fimbriae are also rod-shaped or bacillus. All right, the third structure looks like, we're calling it the S layer and the glycocalyx. Glycocalyx is the old-fashioned term for it, which is why that's what you'll see it on the handout that I gave you. We're calling it the S layer now because it's more commonly known that more bacteria form slime layers. So the glycocalyx, this is also a sometimes. So most bacteria have some version of this, but not all. And it is composed, structure first, then function, just like in Bio1, right? Structure first. It's composed of polysaccharides and polypeptides, so sugars and proteins. And I'm not giving you their specific names because it's different in different bacteria. But what we need to remember about this particular structure of sugars and proteins is that it's very sticky. Right? So it's going to help with adherence. That's its main job, is to help with adherence to, again, inhospitable places. If it is present all the time and we say well organized, and I would add thick to that, then we refer to it as a capsule. Most bacteria that are respiratory bacteria have a capsule because it's difficult for the organisms to stick. So this capsule can function for adherence. It also helps prevent dehydration of the bacteria, obviously. And it also inhibits phagocytosis. So I'm going to put a down arrow for inhibiting, and I'm just going to, I don't think I have enough room to write, sorry, to write phagocytosis. Oh, right up here. I don't have enough room. So those are the three main functions of if it's highly organized and it's a capsule. That means every time we stain, we see it, it's always there, if the bacteria possesses it. If it's not so highly organized, if it's thin, if sometimes it's present, sometimes it's not, if there's space between it, it actually looks like now they're showing the slime layer to be much thicker than a capsule because there's gaps in it. Then we call it an S layer, which stands for slime layer, which I like to say slime layer because it sounds cooler than just S layer. And it's same, its main function is for adherence. And what we're finding is many bacteria stick to each other. So the first bacteria sticks to the surface because it has one of these slime layers on it. I'll just draw it kind of jagged. And then what happens is other bacteria can adhere to this bacteria. And we can make whole, I like to think of it like a rainforest, right? We can make these whole complex layers of different bacteria. They're called biofilms. And it's turning out that we originally looked for them and found them on inanimate surfaces, like say in hospitals, like in tubing, right? That's in a patient. And then we get this complex infection of different organisms in that patient. Now we're finding out they're on your teeth. They're on our skin. They're all over in the environment as well as physically on us. And sometimes they contribute to our good health, and sometimes they obviously contribute to poorer health. So this is a whole interesting area of microbiology that's like really going on, a lot of research going on in that area right now. All right, I think that's enough about the external features of. So this yellow, I would say, was probably supposed to be the glycocalyx. This red layer here that I've drawn, that's going to be the cell wall. And almost all bacteria possess a cell wall, and it comes in 2-3 major different forms chemically. So we're going to talk about that for a long time next. And then this layer that I drew here in black, that's the cell membrane or the plasma membrane. And it is, notice, protected. It's underneath the cell wall, And if the bacteria has a glycocalyx or slime layer, it's underneath that as well. So this is right up against the cytoplasm of the organism, right? So we check this box, glycocalyx. Now it's time for us to talk about the cell wall, which I think I'm going to go back here and get rid of this guy, put him in the trash. No, that's going to be dangerous because I think it's going to clear all of them. So let me just erase this guy. All right, let's try to get through the cell wall, and then we may have to finish this lecture on Monday. Let's see if I can get through all of it. Okay. All right, cell wall. Now, the cell wall is your book, the cell wall plus the cell membrane. They refer to that as, and the cell membrane. They call that the cell envelope. I don't know why that we have to do this, but they use the term, and it's going to show up in your Connect questions. And if I don't put it up here, you'll all just be like, I don't know what that means, which I totally understand, but it's annoying because no one I know ever used that word cell envelope, but whatever. Your book does, so we shall. That should be an A. Okay, so we're going to focus right now on the cell wall portion of the cell envelope. And all bacterial cells, and because it's an introductory microbiology course, I am going to say all bacterial cells. There are very few that lack this component. The main important component structurally to the cell wall of all bacteria is something that we call peptidoglycan. And this peptidoglycan, we must know about its components, how it's made, okay? Its name tells us, right? It's made out of peptides, which means short chains of amino acids. And I will frequently do this, AA for amino acid. And of course, glycan implies glucose, which it does, modified glucose. Two glucose modifications. One of them is known as NAG, which stands for N-acetyl glutamic acid. And the other, wait, no, and I can't even, really? Glucosamine, sorry, N-acetylglucosamine. That didn't sound right. So N-acetylglucosamine. The other's an acid. And NAM, which stands for N-acetylmuramic acid. Why am I telling you this? Because this is going to be important for how the peptidoglycan is actually assembled, okay? So I'm just going to abbreviate these as NAG and NAM, and I'm not going to be able to draw them as beautifully as they are depicted in your textbooks. You can take a look at the real pictures there. I'm going to erase this so I have a little more space to write and draw. And just because, again, we need to know the structure of peptidoglycan. So how this peptidoglycan gets assembled in bacterial cells, since these are glucose molecules, I'm going to roughly draw them like this. Right? They're modified glucose molecules. And what happens is, I didn't say they were going to be great glucose molecules. I said they were going to be glucose molecules, right? So that kind of looks like glucose, right? And they're going to alternate. So this one's going to be NAG. This one's going to be NAM. This one's going to be NAG. This next one's going to be NAM. Okay, so they alternate as they're assembled. This. Don't have quite that much room. And I'm just putting M's because I'm getting tired of writing them all out. So first step of the construction is to make these chains that are alternating nag and nam. Okay. Then the chains are joined together in a layer or a sheet. Like this. I'll use a green, not between each of them, but every so often they're joined to another chain. So I'll draw this one over here. Yeah, my drawings are getting worse, sorry. Oh. Okay, you know there's one down there, right? And it's the same. This one's G, this one's M, this one's G, this guy's M. Right? And the green, these are some peptides, some protein that are attaching the chains together. Okay, so the chains are joined together in a sheet, and I've only drawn 2 of the sheets, but it actually goes kind of like this. So the next sheet would be attached here and above and below. Clearly I don't have enough room for that. All right, so I'll draw one more out here like this. Oh. Right, and again, this would be nag, and it alternates with uramic acid, right? And these, again, are the peptides that connect the sheet. So this is the example of the sheet. Now, these sheets can be stacked in layers. So the sheets can be stacked on top of each other, but, important but, there's a spacer between the sheets, and that spacer is made out of 4 amino acids, always the same 4. It's called a tetrapeptide. And this, because I am not much of an artist, is not going to be as good. But if you look, looks like everyone's too lazy to draw this, even graphic design artists, right? Sticking up, I think it actually attaches to the peptide, to the cross-linking peptides. So this is the tetrapeptide here in yellow, right? And then that's going to be able to be an attachment for another layer on top of this, right? Which I don't know how to draw that, so I'm going to stop the sharing, right? So it would be like if one layer, I'm trying to think of a way to do this, right? So if this, like if this index card is a layer of peptidoglycan, and then there's another layer on top of it, the tetrapeptides are my thumb holding the 2 layers apart, right? And then there might be another layer on top of that. So that structure is so important. I mean, I went through all that bother. Oh no, oh, please don't be gone. Oh, they're gone. Oh, I didn't know that. Okay, well, they're gone. So that structure is so important. Hopefully you'll watch it on the video and then you'll be able to see it again. So that structure of peptidoglycan, so let me rewrite this again, cell wall. Right, that structure is super important. And remember, most bacteria, if not all of them, this is a beginning microbiology class, I would say all bacteria have peptidoglycan somewhere in their cell wall. Now, there are different configurations of peptidoglycan to construct the cell wall. One cell wall type is called a gram-positive or a gram-negative cell wall. The gram-positive, gram-negative cell wall type. It's capital G because the gentleman who designed this stain to distinguish these two cell types, these cell wall types, his name was Hans Christian Graham. So we make it a capital G. A bacterial cell that has a gram-positive cell wall has many, 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 many thick layers of peptidoglycan outside of their phospholipid bilayer. So the surface of the bacteria has this thick layer of peptidoglycan, plus underneath that then this is the cell membrane, right? Phospholipids. In addition, sticking out of the gram-positive cell wall type is this acid called teichoic acid. Make it red. TEICHOIC. Teichoic acid. And it comes in regular and lipo with a lipid on top of it. Okay, that's one cell wall type. The other major cell wall type in the gram-positive, gram-negative cell wall type is the gram-negative cell wall. Oh, sorry. And that gram-negative cell wall is different, right? It has phospholipid bilayer on the outside, almost like a eukaryotic cell would have. These are supposed to be phospholipids, right? And then under that, it has a few thin layers of peptidoglycan, 2, maybe 3. And then the actual cell membrane. So in the case of the gram-negative cell wall, right, important molecule sticking out of this outer membrane is called LPS, which stands for lipopolysaccharide. LPS. See how that's different? This is different from this guy, right? So what that's going to mean is that when we apply a stain, and in this case, it's going to be 2 colors, 2 dyes, it's a differential stain, it can tell us the difference between this kind of a cell wall and this kind of a cell wall. And in these cells, we would refer to this portion, right? That's the cell wall portion on a gram-negative cell. And this membrane here, they refer to it as the outer membrane. It's usually as much designation as it gets. And they're going to this outer membrane of phospholipid bilayer is going to react differently than an outer layer of solid peptidoglycan. This particular, while we've spent so much time on this particular cell wall type, is super important because you treat these with different antibiotics. Now, another type of cell wall, variation, is what we call the acid-fast cell wall. And that one is peptidoglycan plus a very, very waxy acid called mycolic acid. It builds up and forms this very waxy structure that's very resistant to water. And why we're interested in this kind of cell wall is because a major bacteria causes major disease, Mycobacterium genus and species tuberculosis. or Mycobacterium leprae. That one causes leprosy. This one causes tuberculosis. They both have acid-fast cell walls. So again, this is a stain that we're going to apply that is going to distinguish or tell us the difference between basically these two bacteria and everyone else, between types of cell walls. And I probably listed on the handout, but just super, super briefly, not expecting you to know the details of some atypical cell walls. There are bacteria that we call L forms. They don't actually have a cell wall. They're usually bacteria that have lived with an organism for a very long time and they stop making their cell wall. And the mycoplasmas. They have a cell wall that is made, they don't actually have a peptoglycan in their cell wall, but what they have instead are steroids to help maintain the shape of the cell membrane. You don't have to worry about these so much, just have to mention them. All right, so I am going to stop.